today the lecture is going to be about chromosomes and the cell nucleus, or chromosomes in the cell nucleus, is another way of putting it. And if you remember from last week, we talked mostly about mitosis and cell chromosomes during cell division. So the nucleus, in most organisms, the nucleus doesn't exist anymore. The nuclear membrane fragments up, and there's no more nucleus. But if you remember the cell cycle, there was this small M phase, and then the rest was G1, S, and G2 phase. And that picture of cells I showed you where sometimes you saw the chromosomes as individual threads, but then there were all these other cells, these interphase cells, where the chromosomes don't really seem to be doing much. But actually, today's lecture will be more about the interphase stage and how chromosomes and the nucleus are interacting during this interphase stage. Even though the chromosomes aren't moving around and being divided, they're still quite active, and it's still a very dynamic stage. It's just not so visible with the naked eye, or it's not so visible in a microscope. But before we get to that, there was a question from last week that Rusan asked me. So the question was, if we have, let's say these are two spindle poles, and if we had a chromosome here in the middle, between the two spindle poles, that is connected to both sides like this, so it's closer to one side than the other side, why does it end up in the middle of the spindle? Why doesn't it just stay here? Because it's being pulled from both sides, and if these microtubules are pulling on the chromosome, it should be enough to pull these two kinetochores apart, and that should be enough to satisfy this spindle checkpoint. So why, if the chromosome is here, what would be causing it to move in that direction? And that he asked me that last week after class, and I didn't quite have a good answer. But I did some reading, and it turns out that one of the main things that is pushing chromosomes away, pushing chromosomes toward the middle, is that they are actually interacting with other microtubules of the spindle besides the ones that they're attached to. So chromosomes are definitely attached by these microtubules to these spindle poles. So if I drew it a little bit bigger, so here's the two sister chromatids, and each kinetochore is attached to microtubules that are going to the spindle poles. And they're being pulled apart right here. But they're also interacting with these microtubules. And instead of being pulled toward the pole this way, their interactions with these microtubules are actually pushing them away. They're being pushed away from these spindle poles. And when people saw this in large cells, so if this ever became unattached, or if they used a laser to cut this microtubule and that microtubule, the chromosome would actually just get pushed away like it was being blown away by some wind from this pole. And they called this actually the polar wind for a while because they didn't know what that was. So anyway, the point is that these chromosomes, they experience a force that's proportional to the distance from this pole. So that tends to push them toward the center here. And then if it keeps pushing them, then they get closer to this pole, and that same force starts pushing them back toward the middle. So it's not this pulling force on these chromosomes between these poles. The main point of that is to increase the tension at this kinetochore and tell the entire spindle that this chromosome is ready to divide. But the thing that's bringing it to the middle is actually this other force where it's being pushed by these microtubules. And so if the force that's pulling this kinetochore to this pole, remember these microtubules have a minus end here and a plus end here in the middle of the spindle. There's a minus end directed motor that is walking towards the minus end. That's localized here at the kinetochore. But actually, at the rest of the chromosome, there are plus end directed motors that want to walk toward the plus end of the microtubules. And I told you last time that there were that these minus end motors were called dyneins, and the plus end, the ones that want to go to the plus end, are called kinesins. And some kinesins are actually bound to the DNA, and they walk toward the plus end of the microtubule, and those are called chromokinesins. So there are chromokinesins that are sitting on the chromosome that are trying to walk toward the plus end of the microtubule, and that's what is pushing the chromosomes toward the center. So it's not these microtubules necessarily that are pulling it to the center, but rather it's getting pushed back and forth until it finally finds a balance there in the middle. Okay. So that kind of dynamic interplay is very important in cell division, but it doesn't really have much to do with this kind of with this stage. Okay, this is an interphase cell. Okay. So I've shown you this picture many times before, and hope we can start recognizing some of the features here. Does anybody remember what this region of DNA would be called? Anybody? So.
Euchromatin.
Yeah, right, yes. Yeah, this sparse, not very dense part is called euchromatin. And then what about this part? Anybody remember that? Yeah. So, right, so in this one we can see euchromatin and heterochromatin. We can also see a few other things in this picture. And then there's this membrane. So I'm going to talk about some of the other things that we can see. The first thing to notice is that this interphase nucleus, unlike when chromosomes are ready to divide, it has a pretty regular circular shape. It's not a perfect circle, but it is circular enough that it's definitely bounded by this membrane. And if we look up close, we can see that it's kind of, I hope you can see that it's kind of like the way I drew it last time. You can see that the DNA is packed up close against this inner membrane, and then there's this gap, and there's another line on the outside. And this is because this nuclear envelope that surrounds all the DNA inside the nucleus is actually a double membrane. So if we look at just the outer leaflet, we have this structure that's called, this is a lipid bilayer, or a phospholipid bilayer, which consists of actually two leaflets of different molecules that are just drawn in schematic right here. The actual structure of these molecules is not very important, but what is important is that, like all membranes inside the cell, they have a hydrophilic part on the outside. So these are charged molecules that like to be dissolved in water. And then they have an oily hydrocarbon center. And then on the other side, there's another hydrophilic part. So it's hydrophobic in the outside, so it's kind of like an oily area in the middle, surrounded by an area that likes to be in water. And that's how this entire membrane can exist in the aqueous, the wet environment of the cell. But of course, the nuclear envelope, the entire nuclear envelope, is not just one of these, but it's two of these. So it's two lipid bilayers together with a little gap in between them. And the gap is somewhere around 10 nanometers in width. But that's not the only thing going on inside the nuclear envelope. Like I told you last time, the nuclear envelope also has these holes in it, these discontinuous holes that are covered with these pores. And at the site, so it's not really drawn here very well, but at the site where the nuclear envelope contacts these pores, these molecules actually bend around So there's a continuous link of these hydrophilic heads of these molecules around there. So that molecules of the lipid bilayer over here can eventually diffuse and get to the other leaflet. And these nuclear pores are very important, and I'll be talking a little bit about them later. But they're the only way for things to get from the inside to the outside without breaking the entire nuclear envelope like happens in cell division. Okay, if we take a closer look at the nuclear envelope, we can see that not only is it, it's not just this nice, clean, self-contained double membrane, but I don't know if we can already see it here. So there's a lot of other membranes in this cell, right? We're looking at just one little part of the cell. Does anybody know what this is? This? Mm-hmm. Yeah. These are mitochondria, and also they consist of many membranes. There's probably other things here that are not mitochondria, but they're also full of membranes. And then, maybe we don't see it in this picture very well. But in the next few pictures, I'll show you another aspect of this. So the nuclear envelope doesn't just exist as this two perfect spheres, one inside the other. The outer leaflet actually comes away and goes into this other structure, this other membranous structure in the cell called the endoplasmic reticulum. And that is a big, very large network of membranes that pretty much fill up the entire cell. It has a few different functions inside the cell. It's one of the places that the cell stores most of its calcium. So if the cell needs to release a lot of calcium, that comes from inside the ER endoplasmic reticulum. And it's also the site of protein translation. So for proteins that need to be inside membranes, they have to be, those proteins have to get inserted into the membrane here at this endoplasmic reticulum. And this, although people thought for a long time that the ER and the nucleus were connected, this picture really shows very clearly that they're connected, because this is the inner nuclear membrane down here. So this would be the inner part of the nucleus. And one thing that can be really seen very clearly is this outer nuclear membrane comes apart into this kind of tube structure that comes out and eventually turns into this endoplasmic reticulum. All these little dots that you see here are the sites where proteins are being translated and inserted into the membrane. Okay, so but those EM sections, these electron micrograph sections, they can give you kind of a distorted view, because you start thinking in kind of two-dimensional terms, and you may think that a mitochondria really looks like this, that it really looks like this kind of sausage-shaped thing with a lot of membranes inside it. But actually, they don't really look like that. They're really big tubes that go throughout the entire cell. And so we should be thinking in 3D as much as possible, and realize that these electron micrographs that we see, although the resolution is very good and we can see very small details, it's really just a two-dimensional slice, and we shouldn't think just in those terms of 2D. So for one example, so we need to use other techniques to understand the structure of nuclear components at a 3D level. So a little bit more about the nuclear pores, because they're quite important for how chromosomes function in the nucleus. Nuclear pores, they're not just little holes in the nuclear membrane. I drew this kind of structure that's embedded in the membrane, but it's much more complicated than you might think. They're actually little micro machines that have a really important job of controlling what can come in and what can come out of the nucleus. So this is a kind of cutaway, sort of semi-three-dimensional image of what the nuclear pores look like. Now you can see what Charlie poorly been describing as the continuity of the inner and outer nuclear membranes in the entire nuclear envelope, and how the nuclear pore, there's proteins on this part of the nuclear pore that actually cause the membrane to bend like this. And so that's how these two membranes get their continuity. So it's really just one membrane, not two membranes. But you see it's got this really interesting structure. There's this kind of basket-looking thing that projects into the nucleus, and then there are these filaments that come out and stick into the cytoplasm. Let's see. Right, so this is just kind of a cutaway of what it looks like. Now, it's not just a single protein. The nuclear pore has a complex of many, actually probably at least 100 different kinds of proteins. And we know that there are, these proteins have particular locations inside the nuclear pore because we can use antibody staining. We can make antibodies that recognize only one of the many, many proteins inside the nuclear pore. I used some advanced microscopy to show, for instance, that some of these antibodies recognize the outside of the nuclear envelope, and some of them are recognizing kind of the inner part of the nuclear envelope. So if we have an antibody that binds to one of these filament projections that come out of the nuclear pore, then it will localize here, kind of on the outside of the nucleus, whereas if we have other antibodies that bind inside the nuclear pore, they will bind right at the surface of the nucleus. So it's by doing a lot of this kind of antibody staining and doing antibodies with electron microscopy that led to this sort of picture of the nuclear pore. So these are really big. If you look here, this is a scale in nanometers, and the total channel size is somewhere from 20 to 30 nanometers, which is big enough. It's bigger than most proteins. So you might think most proteins can just come in and out. And in fact, any protein that is less than this size, so 40 kilodaltons, I don't know if you're familiar, do you know what this unit, what is a dalton? A dalton is just, so a dalton is the weight of one hydrogen atom, pretty much, or it's one proton. So for instance, a carbon atom is usually 12 daltons, right? Because it has 6 protons and 6 neutrons in it. So if you add up all the atoms in your protein, you will get a total number of how many daltons it is. So proteins have a lot of carbon, they have a lot of nitrogen, a lot of oxygen in them. If you add them all up, you'll get some number, and it's usually in the thousands of daltons, so it's kilodaltons. 40 kilodaltons is about is a kind of smallish protein size. So if you made a list of the sizes of all the proteins in the cell, 40 would be just about somewhere in the middle, I think. Maybe a little on the small side. And I guess this should be greater then, because anything that's less than 40 kilodaltons can come in and out very easily. But the nuclear pore can restrict the flow of things that are greater than that. And how does it do it? That was a big mystery for a long time. How does this nuclear pore prevent things from coming in and out? Because the pore size should be big enough that any protein should be able to go in and out easily. But only really small proteins can get in and out easily. And so the way it works is that the interior, the pore size, it's not actually open. It's not actually a hole in the nucleus where anything can come in and out, but it's full of these what are called disordered proteins. So they're very these proteins don't really have much structure. They don't bundle up into a nice protein shape. Instead, they kind of stick out, and it's kind of like spaghetti or noodles sticking out into the middle of this nuclear pore. Another model that looks kind of like this. So there's this nice basket which is made of really nicely structured proteins, and then the interaction between the membrane and the nuclear pore also has really nicely structured proteins. But the middle is just this really disordered mess of, it's almost like a gel, is how people have modeled it. And any protein that tries to get in just gets stuck inside this gel, and it's more likely for it to come out again than to go all the way through. So for a protein to randomly go all the way through, it will take a lot of time for it to just randomly make its way through this gel. But somehow proteins do manage to come in and out. They can do that if they are helped through by a special, there's a special protein that can help carry them through. So if proteins are actively brought in or actively brought out, they can pass through the nuclear pore. But without this active help, they cannot go in or out of the nucleus. So the next part of the nucleus that we need to consider is something that it's not really obvious from looking at the kind of EM images I was showing you. So you can't really see this next thing in this image. The electron microscope can show you the membranes very well, but this kind of electron microscope can't show you the nuclear, this other thing called the nuclear lamina. The nuclear lamina is what's kind of indicated here with this red outline, and it's only on the inner side. There's no nuclear lamina on the outer side. What the nuclear lamina is it's another meshwork. It's another kind of protein that looks kind of like this. So using a different kind of electron microscope, and here we're looking at the inner face of the nuclear envelope. This is another structure made of proteins. These proteins are called lamins. So the nuclear lamina protein that makes up the nuclear lamina is called lamin. And probably every time there's a word that ends in it's usually a protein, because most proteins end in. And these lamin proteins are a type of protein known as an intermediate filament. It doesn't really matter that type of protein, but what's important is that they can kind of assemble into these filamentous structures, and they can assemble both head to head or head to tail, but they can also orient themselves in kind of this kind of meshwork. So they can bind each other's, the heads of two of these proteins can bind each other, but they can also bind sideways like this, so you get this nice kind of mesh that lines the entire interface of the nuclear membrane. And what's thought that the nuclear lamina does is it actually provides this rigid structure, this rigid shape, which is one reason why the nucleus inside the cell can be so nicely circular. The cell is, of course, moving all over the place and getting all the organelles inside the cell can be moving, but the nucleus maintains a pretty nice shape, mostly because this nuclear lamina resists compression forces. So this is one thing. And another function of the nuclear lamina is a lot of the DNA specifically binds to the nuclear lamina, and it provides this kind of a landmark. There's nothing else inside the nucleus, so the only thing that there is in there for DNA to grab onto is the rim, is this outer edge of the nuclear envelope. And so the lamina provides just a huge surface for any DNA to grab onto to kind of anchor itself if it needs to be anchored. And we'll see that there's some times where it needs to be anchored. So how we know that the nuclear lamina provides rigidity is actually in cases where the nuclear lamina can be disrupted And there's a particular syndrome. It's a very rare syndrome, thankfully a very rare syndrome, but it's called progeria. So the syndrome was known for a very long time before the causes were known. What progeria is premature aging. So children start to show signs of aging, and they would only live into their teens at most, usually. It's quite rare. Maybe 1 in a few million people has this. But the main genetic lesion in some forms of this progeria is a mutation in the lamin protein. So this lamin, this meshwork, cannot get made correctly. Actually, the lamin protein is too big, so it can't make this proper meshwork. And if you look at the nuclei from these patients, you can actually see that the nucleus really loses its nice rigid structure and makes a lot of all these indentations. Probably where the microtubule cytoskeleton in the cell is pushing into it, or some organelle is pushed into it, or maybe even another cell has poked in and pushed the cell membrane inside. These nuclei just don't have this rigidity to withstand these forces. It's not exactly known why. Why should this problem with the nucleus lead to premature aging? But actually, nuclei in natural aging also show this kind of progressive loss of integrity. It's thought that it's probably some effect on the expression of genes, actually, and not so much that the nucleus is not rigid anymore, but this loss of rigidity leads to a loss of organization of chromosomes inside the nucleus, and that can lead to things like premature aging. And in a little bit, we'll see how the lamin helps organize chromosomes inside the nucleus. The last little thing, the last aspect of the nucleus is a small structure called the nucleolus. Nucleolus just means little nucleus, because it's like another nucleus inside the nucleus. And if you remember back to this image I keep showing you, there were a few other structures that we didn't really have a name for yet. But you see that this region looks kind of different, and this region looks kind of different. And you can see also that it has some heterochromosome around it, and this one kind of does too. So these are nucleoli, or that's the plural of nucleolus. And they were, you don't need an electron microscope to see them. They're actually visible with quite, not primitive, but even 100 years ago, microscopes people had would let you see this nucleolus inside the nucleus. So before people knew what the nucleus was, they just thought, okay, this is the center of the cell. It's like the kernel of the cell, so we'll call it the nucleus. And then there's a little nucleus inside it, but nobody knew what it was. People realized what the nucleus was straight away at the beginning of the 20th century. But it wasn't really until the 1960s where people could do some biochemistry and fractionate the contents of the cell before people figured out what the nucleolus was and what it was doing. When we stain all the DNA inside the cell, usually the nucleolus is dark. And that's a hint of what's going on in the nucleolus. It actually doesn't contain much DNA. It only contains a little bit of DNA. What really it contains is, as it says up there in the title, it contains mainly RNA. So what the nucleolus is it's a site where the ribosomal RNA and some other kinds of structural RNA are getting made. And they're really getting made in huge quantities. So the ribosomal RNA is a large RNA molecule that makes up the ribosome, and that's part of the machine that takes messenger RNA and turns it into protein. If I'm speaking gibberish to you, if you don't know what I'm talking about, please ask me a question or come up to me later. I guess there's some bits of biology that some people may not be familiar with. Anyway, all we need to know is that this location in the nucleus is where these RNA particles are getting transcribed. And because there's so much RNA and not much DNA, it just kind of collects around the site. It collects in a, what is it? It's kind of like, it's like an oil droplet inside the nucleus of, inside the nucleus. If you have like an oil droplet in water, it kind of tends to collect together. And it's thought that that's the same principle. That's why all the RNA that's being transcribed here tends to collect around the site on the chromosome where that RNA is coming from. So really, the nucleus is just a cloud, the nucleolus is just a big cloud of RNA. It's very important RNA for the rest of the cell, it's probably no different than any other, what's happening in any other gene. So in any other gene on the chromosome, you're transcribing it and you're making RNA. It's just you're not making very much, and the RNA kind of diffuses out and leaves after a while. But the nucleolus, there's so much RNA being made here that it doesn't all have a chance to leave right away, and it collects in this very large bubble around that gene. So I've talked about various structural components of the nucleus, but this is a chromosome biology class, not a nuclear pore or nuclear lamina or nuclear membrane class. So we have to go back to the chromosomes and how do the chromosomes fit into this picture. So this is kind of the image that a lot of people that I had when I first started studying this. This is the nucleus, and this is the nuclear envelope, and this is the nucleolus here, maybe, and then this might be some other part of the nucleus. And the chromosomes are like this. They're these kind of spaghetti-like or udon-like, ramen-like strands. But like I told you last time, this is not the way chromosomes are really organized inside the nucleus. And of course, the theoretical considerations are already enough to tell us that chromosomes couldn't be distributed this way in interphase, because then when they tried to condense, they would get all tangled up. But we have actual direct visual evidence that chromosomes are not organized this way inside the nucleus. And that comes from a technology that lets us label each chromosome in a different color. Okay, so there's this technique called spectral karyotyping. It's a complicated word. So the karyotype, if you remember from earlier, the karyotype is just this. Remember I showed you my own chromosomes and what they look like, and they have these banding patterns. And we can tell which chromosome is which by the actual banding patterns that they have. But spectral karyotyping is doing this not by looking at the banding patterns, but rather by looking at the color of chromosomes after we label them with specific DNA probes. So for instance, we take DNA that is mostly found on chromosome 1 and we label it with a certain dye, in this case some yellow dye, and then if we take this probe DNA and put it in on our cells, then chromosome 1, maybe it's that one, will get labeled in yellow. Likewise, we take DNA that is highly enriched on chromosome 2 and label it red. We actually have to use many colors that are only very slightly different from each other and use some computation to assign these numbers to these chromosomes. But in the end, you get a different color and a nice kind of automatic sort of computer programs that can take images like this and automatically extract all the data and tell you which chromosome is which. So the karyotype is nice, and this is a very nice way to check your karyotype. But what we're interested in here is the interphase. So this may be a prophase nucleus where all the chromosomes are condensed. And we can see the centromeres here and here. But what's going on in interphase, we can see that it's not just all these chromosomes are kind of interdigitated with each other. But we can see like there's a red splotch here, and a blue splotch here, and a green splotch here. So the chromosomes are really kind of isolated in their own little territories. And we can see that even better once the chromosomes start condensing just a little bit. So here, in this image, the nuclear envelope hasn't broken down yet. So all these chromosomes are still inside this nucleus. So once they condense a little bit, then it really becomes clear that each chromosome is in its own little territory. And this was kind of a surprising finding because it was thought that it wasn't really known that these territories would be so compact. So even though the interphase nucleus, and I keep coming back to this picture, but this was kind of the image that everybody had. So in this interphase nucleus, when we look in 2 dimensions, it looks like it's just, we don't see any division between chromosomes. It's not like there's a chromosome here and a chromosome here. It looks like it's just one big undifferentiated mass of chromatin. But actually, that same interphase cell is really divided very nicely into these chromosome territories. And the chromosomes are just touching each other so intimately that you cannot see any gap between them. But in reality, they're all separate from each other. So instead of thinking of it as this big bowl of noodles, it's really more like this, where each chromosome is kind of on its own, and it's all wrapped up. And if you wanted to get the information inside this chromosome, you would have to just pull on this little piece of yarn and extend it out. But it's not going to get tangled up with any of the other pieces. So the chromosomes are all separate from each other inside the nucleus. And so that finding, the chromosomes have some organization inside the nucleus and they're not all mixed up with each other, that led to some other interesting questions about how chromosomes are organized. And there's really, you can think of 3 different ways, now that we know chromosomes are in their territories. How are these territories organized inside the nucleus? And there's at least 3 different ways that they could be ordered. One is, I kind of talked about it a little bit last time, and that is a general orientation of all the chromosomes. So are all the chromosomes, in some sense, pointing in any direction? So that's what I referred to last time as the Rabel orientation. And in many cells, especially cells that are dividing very quickly, when the chromosomes are pulled toward the daughter cells, they are all pointing in the same direction. All the centromeres are pointing toward the pole, and the chromosome ends are pointing the other way. And that organization is maintained into the next interphase, as can be seen in this cell. If you have a probe that lights up the chromosome ends, you can see that even in these interphase cells, all the chromosomes are still pointing in the same direction. Okay, so this, so that is one way that chromosomes can be organized. But that's only really in cells that are dividing very quickly. So the next one is this radial positioning. So the nucleus, being a sphere, it doesn't have any landmarks. There's no left or right or top or bottom of the nucleus. So you can't be organized that way. But you can be organized towards the center or towards the rim. So then that leads to this question. Are chromosomes randomly arranged? We have 23 different chromosomes in our nucleus, and we have an inside of our nucleus and an outside. And if we just ask the question, if we take a random part of our nucleus, if we take the inside versus the outside of our nucleus, are we equally likely to sample each of our chromosomes, or are some chromosomes more likely to be inside or outside? It's long hypothesized, since people have for a long time been able to label the position of a particular gene on a chromosome and see where that gene is inside the nucleus. Some genes tend to be, some genes have been observed to be mostly on, like, if you look at where a gene is, a lot of times this gene will always be seen at the at the edge, sometimes right on the edge, or sometimes close to the edge. But a certain gene may be always observed there, whereas other genes may always be observed in the middle of the chromosome. So by looking at a lot of this data, people suspected that chromosomes may have a preferred position to be inside the nucleus or outside the nucleus, at the outside edge of the nucleus. And the only thing that could make this organization, since there's nothing in the middle of the nucleus for these chromosomes to grab onto, would be if they somehow were able to bind to proteins like the nuclear lamina at the outside edge of the nucleus. So the idea was that some chromosomes or some parts of chromosomes would preferentially like to bind to grab onto the proteins, this nuclear lamina meshwork underneath the nucleus. But doing it one gene at a time will never, it'll be a long time before we can answer whether any chromosome likes to be inside the middle or not. So there's been a recent interesting paper where this labeling method, so a way of labeling DNA, specifically labeling DNA at the outer edge of the nucleus, has allowed people to measure this kind of inside versus outside tendency for the entire genome. Okay, and the way this is labeled is by modifying the DNA itself with, again, with this methyl group that I've talked about before. So I've talked before about methylated cytosine, which is this one. And mammals like us are usually the only way our DNA is modified is by modifying cytosine. So cytosine can be methylated, and then to get rid of this methylation can be oxidized and finally removed. So mammals have these kinds of modified DNA bases, and it's all cytosine. But other organisms, bacteria and viruses and protists, have all kinds of different modified DNA, right? And one of them is methyladenine. So this is adenine. This is the A of DNA, the A that binds to T. It normally doesn't have this methyl group here. Normally this is adenine, but many bacteria have this kind of methyladenine in their genome. And the way they get it is by a certain enzyme, of course, an enzyme called DAM methylase that adds a methyl group to this position. So we have a sequence of DNA, this enzyme acts on it, and now at each adenine there is this methyl group added. But this is a bacterial enzyme. We don't have it in our cells, and so that's why our cells never have this methyladenine. But people realize if you put this enzyme into human cells or into mammalian cells, then you could mark DNA. You could label the adenine residues of our DNA with this methyl group, and then you'd be able to see where this mark is. And so the clever thing that people did, this lab in the Netherlands, was they took this nuclear lamina protein, the green here. So this green bit is this nuclear lamina, and the orange bit is this enzyme that labels the adenines of DNA. So at the nuclear lamina, so at this meshwork, right underneath the nuclear envelope, there's this enzyme sitting there that labels DNA with this methyladenine. And it's nowhere else in the nucleus. So the rest of the nucleus is free and it doesn't have this enzyme. So now only DNA that is touching or very close to the nuclear envelope will get labeled with this methyladenine. And so you let the cells just exist in this environment for a little while. So cells will just do their thing, and the DNA that's close to the lamina will accumulate these labeling with methyladenine. And here it's a diagram. That DNA will turn red. It doesn't really turn red, it just gets adenine. Then you can take all the DNA out of all the cells. Now you can just extract the DNA. Now you're just doing pure biochemistry. You're not worried about the 3D environment of this DNA anymore, because the 3D structure of the chromosomes has been recorded in the positions of where this adenine is and where it's not. Since we know the entire genome, we can sequence this DNA and we can look as we go down the chromosome, where on the chromosome was it touching the envelope and where was it not touching the envelope. And so for that, this is just a little bit of chromosome 5. We can see that chromosome 5 likes to touch the nuclear envelope a lot here, and then it doesn't really touch here. And then it likes to touch it here, and then it goes away. So we can see that chromosomes have certain regions that like to go near the nuclear envelope, and then certain regions that like to be away from it. But we can go one step better and not just look at the sequence, but then we can actually go ahead and look at that DNA in living cells by with another trick, and that is there's another enzyme in bacteria that normally will cut any DNA that has this methyladenine on it, and this is called the restriction enzyme. This DPN1 restriction enzyme. So this DPN1 enzyme first binds to methyladenine and then cuts it. It will cut the DNA at that position. But if you break that enzyme, if you change it in a way that it cannot cut, then it will just bind to the adenine, but it won't cut anything, and so the DNA will be safe. And then the next step is simply taking this enzyme and labeling it with GFP, a fluorescent protein, so we can see it. So it took some different tries and putting the GFP in different places on this protein. But eventually, what you have is this is DNA here, and it gets modified by this DAM methylase bound to the lamina. So the little red spots are where you would get this labeling. And then GFP bound to DPN1 is now showing you where this methyladenine is. And the question here is DNA that likes to bind to the lamina, is it always there? Is it always the same DNA sitting at the lamina all the time? Or does it have a chance to reorganize? And what the answer was if you look at the next cell division, so the DNA was still labeled based on whether it had been contacting the lamina in the previous cell division or not. So it's a little tricky, but here's let's say this is generation 0, where you start labeling the DNA. So of course all the DNA that's labeled is here at the lamina. But then the nuclear envelope breaks up, the chromosomes condense, the cells divide, and then the nucleus forms again. And then the question is, what happens to the DNA that was bound to the lamina? Does it go right back to where it was, or does it go somewhere else? And then the answer was, it's really a mix. So if you just look at where all the GFP is, now it's really everywhere. It's not back at the lamina. But still, it's quite enriched at the lamina. You can see that there's a big part of it that is still there. But there's also a lot of it inside the nucleus, and a lot of it is around the nucleolus here. So you can see it's, of course, not in the nucleolus itself because there's no DNA in there. But around the nucleolus and at the nuclear envelope is where you have a lot of that DNA that was at the nuclear envelope in the previous cell division. So it's really not that. So these regions that have a high tendency to bind to the nuclear lamina, that doesn't mean that they're there in every single cell at every single time. It just means that they're more likely to bind to the lamina than other parts of the chromosome. So chromosomes have this organization. They do have an organization where genes are more likely to be at the outside versus the inside, but it's not 100%, and it's not guaranteed. So one cell may look like this with these orange genes on the outside, but then one cell over, maybe one orange is on the inside and one is on the outside, and then maybe one of the blues is here and one is here. So, so the answer to this organization question is that there is some tendency for chromosomes to be at the edge, and that tendency does does vary depending on where on the chromosome you are. So you definitely have parts of the chromosome that have a strong preference of being at the outside, but it's not a 100% thing, and that they can really reorganize in the next cell division. Okay, so it's not kind of an all or nothing thing where genes have to be in one position or not. Okay, then so the next kind of organization. So there's this gross radial organization that chromosomes can have. And then the final thing is there any organization that chromosomes have with respect to each other? Okay. And yeah, actually this one, this kind of goes with both. So it kind of goes with the last bit where chromosomes are radially positioned. But it's also by painting individual chromosomes with different colors, we can ask now, is there a tendency for an entire chromosome to be at the outside or the inside? Okay. What I'm showing you here is on the top row there's cells from humans, and the bottom two rows are cells from chickens. Actually, maybe they're all the same kind of chickens, right? Okay. And what people have done is not, what these researchers did, is not label chromosomes based on their number. So it's not that they didn't label chromosomes 1 with one color, 2 with another color. Instead, they chose the seven smallest chromosomes of human, and they labeled them green, and all the larger chromosomes labeled them red. Okay, and what you can see in both humans and chickens here is a tendency for the small chromosomes to be towards the center of the nucleus, and the larger chromosomes to be at the exterior. Right. That may or may not be important, but it probably has something to do with the fact that large chromosomes probably have more parts that can grab onto the nuclear lamina, so they have probably a higher likelihood of being able to grab it. Whereas a small chromosome, if it doesn't get a chance to grab the nuclear lamina when the nucleus reforms, then it's just going to get shoved into the middle by all the other chromosomes. So that kind of doesn't go with this. It kind of goes more with the radial versus interior position. But there is a observation that leads us to think that chromosomes have some kind of preferred neighbors as well. So chromosomes don't. I think what in the nucleus is a chromosome able to interact with? One thing is the nuclear membrane. The only other thing, another thing is the nucleolus. The last thing would be other chromosomes. That's the only other thing in the nucleus. And there is, in fact, a tendency for certain chromosomes to like to be next to other chromosomes. And this phenomenon was first uncovered by people studying cancer, in fact. We'll have another class on this later, but in cancer, it turns out that a lot of chromosomes get rearranged. Sometimes a chromosome will break. You'll have a DNA breakage event by perhaps a gamma ray will come in, or a chemical will attack the DNA and break it. Normally, our cells are really good at repairing broken DNA, and it goes right back together where it was. But if it doesn't get repaired in time, then these broken DNA ends will want to fix themselves in some other way. And one way they can fix themselves is by invading another chromosome and breaking it. Okay. So you get these things where you have translocations. You start off with maybe chromosome 1 and chromosome 2, and you end up with part of chromosome 1 bound to chromosome 2, or connected to chromosome 2, and then the rest of the reciprocal translocation. So chromosomes exchange pieces with each other. And what people found is that when looking at different cancers, the same chromosomes would show up much more often. So, what am I trying to say? They would very often see translocations between the same chromosomes very often, whereas other chromosome pairs, they would not see translocations very often. And that led to the idea that perhaps, since the chromosomes don't know when the translocation is going to happen, those chromosomes where you see a high frequency of these translocations, they probably are already next to each other in the nucleus to begin with. Okay, just a lot of text here, but the main thing to come away with from this is that these translocations are non-random, and in fact, one of the most common translocations is a certain translocation between chromosome 9 and chromosome 22. And chromosome 22 is quite a small protein, so it could be anywhere in the nucleus, but very often it turns out to be next to chromosome 9. And what happens when this translocation occurs is that one gene called VCR on chromosome 22 gets fused to a different gene called ABL on chromosome 9. It doesn't matter what these genes are, what they do, but what happens is that the function of these genes gets compromised. When these chromosomes exchange parts, that will tend to lead to this certain kind of cancer. And so the experiment then is to just try to see, are those genes often next to each other inside a nucleus, or are they not? So by looking at normal cells, so these are not these cancerous cells. These are cells where they have a normal chromosome 9 and a normal chromosome 22. But by looking at a lot of these cells and labeling one gene, I think that was the ABL was the chromosome 9 gene, right? Yeah. So the green is the gene on chromosome 9, and the red is the gene on chromosome 22. And now what you're looking at here is a bunch of sections taken with an optical microscope. So this is the bottom of the nucleus, and you're going up through the nucleus, so it's getting bigger and bigger because you're looking at you're looking towards the middle of the nucleus. And you can see somewhere in the middle of the nucleus here, so it's not attached to the nuclear lamina, but it's just floating here in the middle of the nucleus. You can see those two genes are right next to each other. Okay, and the idea is that the graph is not so important, but it really is just showing that that there's a higher than random chance for those two genes to be together. So you have these genes have this entire three-dimensional space of the nucleus that they could be present in, but they happen to be right next to each other. And that happens more often than you would expect by random chance. So for whatever reason, chromosomes 9 and 22 just like to be together. And the mechanism for that, although it's still a little bit unclear, but it's probably that there are some proteins that bind to chromosome 9, and they also bind to chromosome 22, and it just acts as a kind of glue that eventually links these chromosomes together. Okay, and so the last example of chromosome structure that I'll go over is really my favorite example, and it's kind of a surprising example. If you've already heard of this, then it was kind of in some science news in previous years because it was a pretty interesting finding. And that is that, so I've told you that heterochromatin is normally localized on the outside of a nucleus, right? So there's a tendency for heterochromatin to be bound to the nuclear lamina and to line the edges of the nucleus. And that's true for most cells, most cell types. And here, so this heterochromatin is shown with red staining in this cell. And this is a fibroblast cell. This is a cell from internal connective tissue of a mouse. And you can see that, yes, the heterochromatin is really localized to the outside. The same is true for these guys and for these guys. So the red shows you where is the heterochromatin. And like most cells, so this is a ganglion cell in the nervous system, and a fibroblast cell. They have that on the very outside. However, a certain type of cell in mice has the inverted organization. So instead of having heterochromatin on the outside, it's really on the inside. The blue part is blue part is maybe the most heterochromatic zone. So blue is the most heterochromatic, red is a little less, and green is euchromatin. So, right. So And the cells where this is inverted happens to be the rod cells. These are the most light-sensitive cells in the eye. And it's very interesting. Why should these cells have this different organization of heterochromatin versus euchromatin compared to all the other cells? So it's not just the density of DNA that shows this difference. I talked earlier about different histone modifications. So the tails of histones get modified in different ways to show you, to mark where it's heterochromatin and to condense DNA. And so here, if we look at where the rod cells are, I guess this is the most important one. So histone H3 has this methylation of its 9th lysine, and that's shown in yellow here. And the rod nucleus here has that kind of in the middle, whereas the typical nerve cell has that just in these blocks more on the outside. Okay, and there's just another example where you can see the heterochromatin here in the middle, whereas the heterochromatin is at the rim on the other one. So heterochromatin and euchromatin is inverted in these rod cells. And so, yeah, that was kind of puzzling to people. But then some researchers thought, well, okay, this is what we've seen in mice, but let's look at other animals, because maybe this is just something that's happening in mice. And mice have a lot of heterochromatin on the ends of their chromosomes, so where their centromere is actually at the end of the chromosome. So maybe this is something strange just happening in mice. So they took a lot of different animals. In fact, I don't know if you can read it, but so a cat, mouse, lemurs, rats, deer, rabbit, horse, cow, tenrec, pig, shrew, and a monkey, and a chipmunk. And I just labeled them. Yellow is the normal organization where heterochromatin is on the outside. And blue is this inverted organization where heterochromatin is on the inside. So mice and rats have this inverted organization, whereas cows and macaques, which is a monkey, and horses and pigs, and humans, in fact, also have that organization. So it's not all eye cells that have it, but only certain ones. But the interesting thing is that it didn't really show. You might think, okay, so some animals that are more related to each other have this inverted organization. So mice and rats, they're pretty closely related to each other, so maybe they have the same organization of their heterochromatin. Whereas, well, okay, these aren't really too related to each other. Maybe horses and cows, sure, they're related, and yeah, but actually, when they tried to map the different organizations onto this phylogenetic tree. So here, this blob is the inverted organization where heterochromatin is in the middle, and these are where heterochromatin is on the outside. It didn't line up at all. In fact, things that are really closely related, like these three rodents, you have inverted, normal. Here, two different kinds of monkeys. Well, different kinds of primates. The lemurs have it in the middle, and the macaques have on the outside. So they didn't really show phylogenetic grouping. And can anybody guess? There was a different grouping, though, that made sense of this pattern. So that made sense of why some animals have heterochromatin on the outside, and some animals have heterochromatin on the inside.
Was it the nocturnal?
Yes. Yes, that's right. Yes, you got it. So that's exactly right. So it didn't correlate with which animal was related to which, but it correlated very well with whether an animal was nocturnal, whether they were active at night, and whether they really needed their rod cells to get every last photon from the night environment, or whether they really didn't need their rod cells and they got plenty of light. So, okay, I don't know which kind of lemur they used. I don't think all lemurs are nocturnal, but I guess they happen to use a nocturnal lemur here. But definitely cats and mice and rats They're nocturnal, and chipmunks, I've only seen them out in the daytime. So I think I believe this. And they did their checks really well to show that that was true. And it didn't stop there. They in fact did some very interesting modeling with light, and they showed that the heterochromatin will actually change the refractive index of the cell enough so that light that comes through a cell with heterochromatin on the outside will get scattered and will not penetrate as deeply. And so there will be a lot of light lost. Just a little, well, not a lot. Actually, it's a very small effect. But it's enough, having a heterochromatin on the inside concentrates enough refractive index so that it acts like a little lens, and you get a nice, stronger penetration of light, so that these nuclei act like little lenses, and they're exactly where you want them in the rod cells that are supposed to be most sensitive to light. But that advantage you get from seeing a little bit better in the dark is, I guess, even though it would be nice for us too, it's not, it's outweighed by whatever advantage we get of having this normal heterochromatin on the outside kind of organization of our nucleus. So if it didn't matter at all, I guess we would all animals would have this sort of organization, because why not? Why not see a little bit better at night? So it obviously matters for something, but it's probably changed and flipped in many different lineages depending on whether the animal was nocturnal at the time. I'm sure chipmunks started out as nocturnal, but they moved to a more diurnal lifestyle and then flipped back. So it's probably very, very flexible, and yeah. So we're stuck with the daytime organization, but it's probably doing something good for our eyes. Okay. So the very last thing, and I will kind of leave with the last bit of nuclear organization. I told you that chromosomes do not take on this kind of bowl of ramen organization, and they're existing in their own separate territories. But there's one time where they do actually look like, maybe it's a little hard to see, so go with that, where they do look like that, and that is in the cell division called meiosis. And that's what we're going to start covering next week, so I'll just end with it on this time, at the end of this class. There's one more kind of large-scale organization of chromosomes, which is thought to be important for meiosis. And what happens is that all the telomeres, all the ends of chromosomes, are brought together at one location on the nucleus. So I've said before that there's no landmark in the nucleus. There's just this. There's the outside and the inside, but there's no place. There's no left or right. But here in meiosis, it would seem that there maybe is one privileged region, because all the chromosome ends are all brought together at that location. And this organization of chromosomes is thought to be very important to allow chromosomes to meet each other. But I'll just leave you with that picture, and just to kind of remind you that even though I may say that this bowl of ramen organization does not exist in cells, there's always exceptions in biology, and there's always going to be some kind of weird cell type or some organism that does the opposite of what you expect. And so in meiosis, you do get the opposite of what I've been saying, and you do get a situation where chromosomes can entangle with each other, but meiosis has some special ways of dealing with that as well. So yeah, I will stop there, I think, and if anybody has questions, I'll ask them. Yeah, or if anyone has any questions now, that's fine too.